Great, thanks. Let's move on to our second talk. Our second speaker is Chao Ye. Chao is a PhD student from China, is a fourth-year student at the Department of Biochemistry. His current scientific interests lie in microbiology and systems biology. He used to work on viruses and yeast before joining James Lock's lab at the Sainsbury Laboratory. And if you haven't seen the Sainsbury Laboratory, it's really worth seeing. It's one of the nicest buildings in Cambridge, overlooking the botanical gardens. In his spare time, Chao enjoys playing basketball and studying the history of humans and of science. His title today is Clock and Cell Division Pulses in Bacteria.
Hi, everyone. Thanks for coming to my talk today, and it's really my pleasure to present my work to all of you. Actually, I've studied here in Fitzwilliam College as an undergraduate for 4 years, and now I've spent another 4 years here as a postgraduate, so it's really a pleasure to come back again. So today I'm going to show you a story about how I studied an interesting phenomenon related to clock and cell division in bacteria. So because of the sun and the self-rotation of Earth, we have 24-hour day-night cycles every day. Nowadays, we often use our clocks, or use mobile phones to keep track of our time. But in fact, over the millions of years of evolution, nature has already created an internal pacemaker in our brain called the circadian clock. In mammals, such as humans and mouse, there's a region in our brain called suprachiasmatic nucleus, and that region actually controls our sleep and wake cycles. And if you track the expression of circadian genes within this region, you can see they pulses up and down over time, and the period is exactly roughly 24 hours. But apart from mammals, many other organisms, such as plants in this case, there's a seedling of Arabidopsis, and also some bacteria, for example, here, the photosynthetic bacteria, cyanobacteria, they also involved circadian clocks. So over the past 4 decades, the molecular mechanism of the circadian clock has already been studied extensively, and I don't want to go into details. But what left for us to study? So actually, the question that we ask in our lab is, how does a clock couple to other cellular processes, especially at a single cell level? So for example, how does the clock couple to cell division cycles from one cell go to 2 daughter cells? How does it couple to photosynthesis, where you convert light energy into biomass? What about metabolism, where you eat food and produce energy? And finally, what about stress response? In our lab, we use a model organism called Synococcus elongatus, which is a cyanobacteria species, to answer these questions. So one way for the cyanobacteria clock to couple to other cellular processes is through sigma factors, which is kind of like a transmitter of the circadian signal from top to the bottom. Over the past 4 years, we've screened a lot of sigma factors in cyanobacteria, but one of the most interesting one is called RPOD4. But before I get into its own expression dynamics, I'm going to show you a movie where we can track the expression of key circadian genes, in this case, KBC, over time using fluorescent protein reporters. Hopefully, as you can see, as the cell divides over time, the expression of circadian genes actually is quite synchronous across different cells and across different colonies. But when we look at the expression of RPOD4, hopefully from the movie you can see the expression of RPOD4 doesn't seem to be circadian. Instead, it pulses, especially when the cell divides. So how can we extract data from it and understand what's actually going on? What we do is to extract the fluorescent signals from these movies and convert them to actual data. So what I'm showing here is as the cell grows over time, become longer and dividing into 2 daughter cells, either in subject day or subject night, the expression of circadian genes, in this case KBC, actually shows a nice circadian oscillations and is quite synchronous between different daughter cells. However, when we look at the expression of RPOD4 in this case, it actually peaks, most of them, at the cell division events. However, you might also notice that at some division events, expression of this gene seems to be quite weak. So what's actually happening here? Does this mean there's a circadian clock effect that's controlling the expression of this particular gene, or there's something else that's going on? To answer this question, we turn to a new technology developed by my colleague, Chris and Sasha. They call that the green mother mission, because you can grow all the mother cells in these thin channels and observe what's going on within these single cells. And actually using this device, we achieved much better signal to noise ratio of RPOD4 in this case, as you show this in this figure, where you can see at every single cell division event, you can get very obvious peaks. However, this doesn't seem to be circadian at all. So what's actually going on? What we next do is actually extract the peak from all these single cell signals and plot them, the mean, against time of the day. We showed that the peak amplitude of RPOD4 expression actually followed a circadian rhythm where it goes up during the subject day and suddenly drops to the basal level and then remain low throughout subject night. This looks quite interesting, but what are the functions does this dynamics bring to? So first, we examined what's the effect of RPOD4 on controlling the division length over the time of birth. As you can see here, if the cells are born as subject dawn, the difference between the wild type and RPOD4 knockout mutants is quite small. But as time goes by, and expression of RPOD4 also goes up, the difference between the wild type and the mutants also becomes larger, and the largest point is close to the early subject night. If we overexpress RPOD4, we see a clear dose-response relationship between the cell length and expression level of RPOD4 controlled by a chemical called IPTG. Basically, if we overexpress RPOD4 more, the cell length becomes longer. In addition, when we examine the clock period of the RPOD4 knockout mutants, we observe that the free running period of the clock actually decreases over time compared to the wild type, which is in orange, and the mutant is in blue. And if we overexpress RPOD4 in this case, the free running period actually increases over time compared to the wild type and non-inducible mutants. So to sum this up, using the single cell time-lapse microscopy, you found that there's an interesting gene called RPOD4 that actually pulses at every cell division. And its expression level is actually modulated by the clock. Finally, the deletion or overexpression of this gene actually can alter the cell size and the clock period. That's summarized in this illustration here. And with that, I'd like to thank my supervisor, my colleagues, and all my collaborators over the past 4 years, and all the funding bodies for my research. And thank you for your attention, and I'm happy to take any questions.
Thank you, Joe. Questions, I can see one straight away. So.
I'm wondering what's the nature of RPOD4? Is it a transcription factor or receptor? Is it only bacteria or also
include? I didn't
do you want to just repeat the question so that the people in Zoom hear it?
So
why don't you repeat it? Because it was technical enough for you to have understood it better than me.
Okay, sorry, I'm too excited to answer the question.
Will you repeat the question?
Yeah, I can repeat. So basically, the question is, what is the nature of RPOD4? Is a transcript factor or a other kind of enzyme that only exists in bacteria or also exists in humans, for example? So the answer is RPOD4 is a sigma factor. It can be considered as a transcript factor because it's an important subunit of RNA polymerase, which is involved in transcribe DNA into messenger RNAs. It's relatively conserved inside bacteria, but it's not conserved in humans. So it's only happened in some bacteria. But it doesn't mean there might be other functionally conserved proteins that might exist in humans that we are working on that to try to identify that.
Thank you. Go ahead.
Yeah.
So humans also have a biological clock, and are there similarities in the human biological clock with the patterns that you're describing here?
Yeah, so basically, throughout the evolution, actually, we think the clock actually evolved multiple times throughout the history. And in humans, the clock is more sort of comprised of transcription and translation feedback loop, which is more relied on transcription itself. Having in bacteria, in this case, and bacteria, they are more dependent on post-translate oscillator. Basically, you rely on oscillation of protein itself. Actually, what's interesting about the bacterial clock is you can take out the proteins, put them into a tube and add ATP, and they can oscillate within that tube without any other substances. So that's a key difference between bacterial clock and human clocks. But there are also studies shown that, such as in red blood cells, which lacks nucleus, people believe there are some ancient protein-based mechanism resides there, and there's ongoing research trying to establish what's the function of that Asian protein-based clock within humans.
Thank you. Chao, can I ask you a sort of general question about what life has been like in the laboratory over the last 18 months? How was it, especially during the sort of periods of closure? Did work continue, or did it impact your work quite a lot?
Yeah, so basically during the first lockdown, which lasted for 3 months, we were not allowed to go back to the lab, which is quite stressful. But thankfully, all the members within the lab, they tried really hard to reopen the lab. And once it's reopened, we kind of get back to normal with like working 3 days a week. And also my supervisor is quite supportive, and he gave me 3 months extension of my PhD, so that actually by now I should submit my thesis, but now I can spend some more time to get back to some more data that I don't have time to analyze during the 3 months period of lockdown. So that is great. And
so you're back on track.
Yeah.
That's great. Thank you very much, Chao, and thank you for sharing your work. Good luck with the finishing up.